So we're running short on time here for our regular program, but I'll let the audience in on a secret. I knew that we were going to get a whole bunch of questions today. I knew that we would, because we did the last time we did this, and I came in with more questions than we could respond to. So I secretly, in advance of our program, I tapped Roberta and Akhilesh on the shoulder and said, "Hey, how would you feel about sticking around for 10 or 15 minutes to do some rapid fire response to audience questions?" And the folks who are asking questions right now are asking good questions. They're engaged, and I don't want to ignore them. I want to tackle as many of them as we can. So you guys game still for spending a few more minutes kind of tackling some of these questions?
I'm good.
Yeah, I think we're good to go. Yep, we're good to go.
Just a few minutes, and then I'll let you off the hook. But let's run through some of these. So, geez, I don't know. You guys can see them too. Do you have any real specific preferences around what you'd like to tackle here?
I actually cannot see anything. Oh, there you go. Yes.
You got it now. Yeah.
Yes. Okay.
I'll throw one out there, and while I'm doing that, if you see questions out there in there that you feel excited about addressing, you just have at it. But let's address that one around process development runs. How many process development runs are generally needed to get a final GMP run conditions defined and fixed? Do we know that yet?
Well, again, it's a question of how well you understand the process. If you're talking of a PPQ stage, your traditional is 3. If you're talking anything before product characterization, it could be more. But end of the day, what you're trying to do is if you have a target process where you are able to deliver the product quality attributes and you have a tolerance window around it, then all you have to prove is consistency. How you prove consistency is 2 max, 1 min. So you are bracketing the max and the min with 2 max, 1 min. That's a traditional approach. However, if you are carrying multiple trains, so say for example, it's a lyo product, and you have 2 different equivalent lyophilizers. When I use the word equivalent, they may or may not be equivalent, because stability was another challenge Roberta was talking about. So with those, if you have 2 max, 1 min, then you have to run another max. So even though I don't think the question is very well defined in my viewpoint, because I don't fully comprehend which part of product or process development they are talking about. But if it's a PPQ related question, then I would say bracket in show consistency and process control. So I go back again to the same feedback of, if you understand the process, if you can monitor the process and show the process is in control, that will give you the number. At a minimum, expectation is 3.
And this, my friends, is why I lean into you two to determine which questions are responses to which questions are gonna provide the most value for our audience. Because I'm telling you, I don't know.
Well, Matt, maybe I can pick up on another set of questions. So Akhilesh kind of touched this a little bit. There are a couple of questions about lyo formulation and other delivery systems, such as inhalation form. So the mRNA technology actually lends well into lyo, and I know there are several companies that are looking into lyo formulation. And so there are going to be questions around capacity and volume that lyo can deliver for the population that you're serving. So larger population, more doses, more volume. Lyo may struggle depending on the capacity there. For smaller populations, smaller volumes. When it comes to inhalation or other delivery systems, those, again, mRNA lends well into that space. So I think those are all of the things that we'll see evolving in the next few years as to what are the multiple formulations and delivery systems that we'll see mRNA vaccines and mRNA therapeutics brought forward. And so there's more to come there, and a lot of opportunity for innovation in lyo, quite frankly, to enable the capacity and quick turnaround for large population volumes.
Excellent. Thanks. Dr. Bhambhani, did you see another question that appeals to you as Roberta was discussing lyophilization?
Yeah, lyophilization, I don't see a question, but ask the questions and I'll tackle it. However, when I go through some of those questions, there is a common theme. And I'll say if there's something to take away from this conversation, something to remember when you're developing mRNA LNP, because those questions are framed in one way or another, they are asking the same question. Um, to be successful for any application, mRNA have to surpass their own challenge, and that is situated at delivery and immunogenicity. You cannot have one and forget about the other one. So when you're talking about characterization, when you're talking about how do you characterize this, what is your CQA, what factors do you consider, how should we scale up, those are the two things you have to take in parallel, your delivery and immunogenicity. I could not stress the significance of in vivo characterization during early development and process optimization till you have the FMP lock. FMP, the final manufacturing process lock. mRNA LNP development is an iterative development process where you need continuous communication from your translation research to your process analytical formulation, the PF as I call it, to manufacturing team. So if you have those, then you may have a right balance of immunogenicity with delivery. You cannot focus on efficacy. You cannot just focus on in vitro characterization and forget about in vivo as I'll develop a process or I'll do a lipid selection. You can select 100 lipids that will give you a good process. But if you do preclinical animal studies, less than 5% would work. So in vivo and in vitro, hand in hand, two sides of the same coin, those are needed to bring a good product to the market. I think that may have answered many of the questions. And if there's a live specific question, please feel free to ask. I
No.
also serve as president for International Society of Lyo and Freeze Drying East Coast chapter. I've been doing Lyo for extensively for many products. So Lyo question, please. I haven't seen any, but go
Yeah,
ahead.
I think we tackled that. I think Roberta kind of tackled that one, and I appreciate you, Dr. Bhambhani, for wiping several questions off with one very valid and thorough response there. So thank you. I've got one here from the registration questions that came in that alludes to something that we talked about prior to going live around supply chain and raw materials. So a question that came in during the registration process around critical raw materials: are you multi-sourcing? And if yes, do you source from more than two? And how you're qualifying the materials in terms of QC testing, like side by side when you're multi-sourcing materials? I'd open. Roberta, sounds like you might have a response to that one.
Yeah, so to be transparent, warp speed and the efforts to deliver the COVID vaccines have impacted all of us, in addition to supply chain challenges that come through the fact that we're in this pandemic. And what has happened as a result is we absolutely have to look for multiple sources for raw materials and test them to demonstrate that those raw materials will work in our processes. So it is an ongoing challenge that we face, and I don't believe we're unique in this. But what we are doing, what it has done, it's challenged, and our team has risen to the challenge, has challenged our procurement, supply chain, quality, technical development, research, all of our colleagues to work very closely together to try and run through these processes of testing materials, working with suppliers to provide COVAs, and even some additional data to give us a greater confidence in the materials we're receiving. Long lead times, things that were taking weeks in the past have now gone out to 49 and 52 weeks. So as you're thinking about your development and your development pathway, there is this moment that's come into place, which has added time in a way that I don't know that everyone recognizes, which is the time to identify, test, and enroll and qualify your raw materials. The timing has increased significantly, and you'll have to come up with creative solutions and ways of working in order to get through those processes as quickly as possible so that it does not slow down your development. And I have to give a shout out to our team who has done a phenomenal job in working through this, but it takes a high level of engagement, a high level of collaboration, and it takes making sure you're very clear on the standard that you're meeting, so they're not falling below that and putting your materials into question. down the road. So just a point of reference, time matters, and it's not necessarily in your controls. You have to come up with creative solutions to claw that back.
Yeah, excellent. Dr. Bhambhani, anything to add there regarding sourcing?
When it comes to sourcing, it's always good to have multiple sources if you can afford to. What I mean by this is you have to show comparability. End of the day, what it boils down to, when I say process is product and you have to build quality into the product, what I mean by that is you can bring multiple sources, but every time you bring a new source, I was using the word CMA, critical material attributes. When we are discussing critical quality attributes, your raw material control becomes a critical aspect of how you are controlling the process. So every time you bring a new lipid source, a new source of cholesterol, DSPC, something else, PEG, that's a good thing because it helps your supply chain. But on the flip side, you have to generate data to show they are comparable and does not impact your product quality attributes in any way, shape, or form. So equivalency has to be established.
Perfect. I'm gonna put another question to Roberta, another registration question that came across. And while I'm doing that, Dr. Bhambhani, if you want to look through the questions that have come in the meantime and see if there are any new ones that haven't been addressed that I can hit you with next. But we had a question in the registration process from Parisa around regulatory affairs specialties needed for a company that is new to mRNA vaccination manufacturing and is in the process of adding more employees with specific regulatory expertise. So she's looking for advice on what sort of regulatory expertise might be more applicable than maybe a generalist in terms of mRNA vaccines. Any thoughts on that? Is there sort of are there sort of some rabbit holes you can go down there that would characterize a good regulatory person for this space better than others?
Yeah, I mean, I think it's less about the mRNA specifically and more about the depth and breadth and understanding that the population and the division of the agency that I think is really critical. With mRNA being relatively new, only 2 vaccines having gotten through licensure and approval, you're not going to get a large pool of specialists focusing in the mRNA space. You will probably run into individuals who've had early interactions with agencies, probably in filing an early IND, some pre-IND discussions, et cetera. But I would say you're looking for someone who really understands the space. In our case, we want someone who understands vaccines, understands the division in the agency, has a depth and breadth in this space and can bring that knowledge forward and is able to take multiple aspects of their prior experience and apply it. So I don't have a specific answer, but when we think about what are those attributes, those are some of the minimum ones I'd be looking for.
Perfect. All right, great. Akhilesh, looks like you picked a couple out that you might want to address here.
They are the top ones. So no particular order, they just happen to be on my screen.
Oh, I know. Listen, it's impossible to follow a particular order at this point. Too much going on.
So this question was from Arunendra Sahare. And no. This is, Lyo often cause LNP size increase despite using cryoprotectant. What is your take on this challenge in cycle development? And this is a common problem. This is something that has been in exploration for almost a decade since siRNA days. There have been a couple of publications on Lyo, even though there is a particle size increase, and the particle size increase post Lyo is probably 5 to 10 nanometers. I don't think in terms of the critical quality attributes, it's changing anything. This is something for us as a community. When I'm using us, I'm actually engaging everyone here who is working on this field as a community to prove, does size going from 70 to 80 nanometer and 80 to 90 nanometer, does it actually make a difference in the biological function? And that comes back to the questions we were talking about before. Biology is the one that you are worried about. Process and manufacturability, you're showing it's in control. So if you have a manufacturing, your product is approved based on efficacy and safety. Your product is not approved based on manufacturing efficiency. Manufacturing, you want to show control so you can have safety and efficacy. So if you can prove it, I don't think that's a problem. The other thing to note is fast freeze, in my opinion, should result in a much lower particle size. Again, this is without running any experiment. In justification, usually LNP should act as nucleating site. When something acts as a nucleating site, you will have a lot more heterogeneity during the freezing process, which will manifest into a particle size increase or osmotic imbalance. So try looking into freezing differences and the osmotic imbalance during the thaw process. Much of the damage may also occur when you're thawing back a solution, thawing as in reconstituting back a solution, because that's when the shock occurs, and a packing defect manifests itself in a particle size increase upon reconstitution. So those are two factors I'll play with to address
Very good.
that increase in particle size. Go ahead.
Perfect. Thank you. Thank you, Dr. Bhambhani. A couple more here, and then we'll let you guys off the hook. But Roberta, a couple that you picked out here is suitable for you. With long lead times for plasmid production, do you see industry moving towards synthetic DNA, like doggy bone DNA technology for mRNA applications? And then there was another one there around how many mRNA vaccines can be mixed for injection. That's probably a quicker one to answer, but I'll throw them both out at you at the same time. And you can pick which order you want to respond to those in.
I'll take the mRNA vaccine mix as the first question. So this is really applicable and apropos in influenza. So when you look at the current mRNA vaccines, it's a single pathogen in each of those vaccines. So they're monovalent vaccines. When you look at influenza, on our pandemic side, yes, our vaccines are targeted to a single strain. When you look at seasonal influenza, however, quadrivalents bring the greatest protection against influenza infection. So when you look at the mRNA vaccines and when you look at the Seqirus portfolio, our portfolio consists of quadrivalent vaccines targeted to give better protection. So our mRNA won't fall below that bar and that standard that we've set in order to bring appropriate influenza protection against the influenza vaccine. So when you think about seasonal influenza, multiple strains of influenza are circulating at the same time. So when 4 strains are identified, 2Bs and 2As, vaccines that incorporate all of those 4 2As and 2Bs, that allows for greater protection around what's circulating in wild type, and also duration of protection using the platform or the modality that you're bringing forward. So mRNA vaccines have to be able to deliver at least to that minimum for us to ensure continued appropriate protection against influenza. Now, other disease targets are going to be different. But the demonstration of the possibility for it to be effective in that space has to be proven in flu.
Mm-hmm.
So that's a critical. It has to be a yes for it to be appropriate for flu. The other question on plasmid DNA, very good question. We know that microbial fermentation, which is the traditional approach today, does have added time constraint into your formulation and manufacturing timeframes. So any innovation in this space that includes synthetic, that will draw that time down to be shorter and give greater confidence in the sequence is going to be critical for success. So we need to look at this space more closely, and I'm sure there's room for innovation in microbial in fermentation to shorten that time. But biology is biology, right? You can't rush necessarily biology, create other challenges. So I would say yes, there are opportunities, but I will also say there's room for both as long as the innovation continues to evolve under the traditional path that exists today.
Excellent. Listen, you guys have been beyond generous with your time, and I need to draw the line somewhere. So I'm going to draw it here because you know why? Because I want you both to come back. So I don't want to overstep my welcome. And judging by the engagement level of our audience, there will perhaps be a part 3. And I want you back for those. So I want to give you each an opportunity, though, to wrap things up here. And if we didn't get to your question, I apologize. But we will, this discussion will continue to go on in many forms, here on BioProcess On Live. Online Live, the Business of Biotech podcast, BioProcess Online. We're always talking about this stuff. So we'll get to those questions another time. But I want to give you each an opportunity to wrap things up with just some concluding thoughts. You've had a taste here now over the course of the last hour plus of the interest level, the questions that have come across from the audience. You know who you're speaking to. Dr. Bhambhani, we'll start with you. Just give us some, give I guess a pithy concluding statement to the attendees.
Sure. I'll start with first thank you to you and the team for pulling the community together on this topic. I think this is just an inflection point. Every company by default, our true North Star is our patient. When we start with a patient in mind in product realization for a given jurisdiction, things become easy. It's not always possible in the early development of manufacturing because many of the questions I'm seeing, people have to realize in the early stages of development, there's manufacturing and control procedures are not in place. Procedures are not validated. So if you adequately address them, the word adequate is important to allow assessment of safety and the risk during the manufacturing process. That means you have to be phase appropriate. The level of detail required by authority goes up as product development increase, but always keep your patient in your North Star in mind and things will just follow. So keep that always in place when you're developing a product.
Dr. Bhambhani, thank you. That's very sound advice. And thank you again for being so gracious with your time with us today. Roberta, we'll let you have the last word, parting thoughts.
Yeah, so Akhilesh hit on a lot of the critical factors, and that North Star is a critical one. We also have patients and public health as our North Star. Being a company that focuses on rare diseases with plasma-based products, and also in a company that focuses on influenza, which is public health, and with multiple platforms. There's a couple of parting thoughts. A lot of great comments and questions. Thank you for the audience for providing them. For Seqirus and CSL, I just want to say that we're really excited about this new location in Waltham, where we are further investing in innovation, further investing in local and in talent in this space. So just keep in mind that as innovation continues to bring forward new opportunities, it shows up in many different ways. When we look at the questions around mRNA, we're at the beginning of that story with our current vaccines that are approved. There's quite a bit of opportunity for innovation in speed of formulation, in characterization of materials, in getting through the regulatory process, in ensuring. There was a plasmid DNA question about how can we innovate in that space. There are questions about LIO. The innovation is just not only in what the sponsor company is doing, but in the entire ecosystem of partners in this. And it is an entire ecosystem. There was an early question about in-house versus outsourcing. And I don't think it has to be either or. There is a hybrid approach. And so you look for what is your critical talent, what is your critical focus that you need to keep in-house, and you determine how you handle the rest. There is no one-size-fits-all answer. It's the right answer that's fit for purpose for your company and for your product and for your patient population to get your product out as quickly as possible to treat diseases, change lives, and improve public health.
Beautiful. Yeah. Yeah. Well, I appreciate, again, the time that you've both given us and the excellent work that you're both doing with your companies. So thank you. Again, very gracious of you to